Hi, good afternoon, everybody. Thank you, Claudia, for the introduction. So my name is Morgane Roux. I'm the Application and Support Scientist for Europe at ATD, and I'm going to present you today the new 2.5 Duplex Assay. Our webinar today, I'm going to show you how the RNAscope technology works, and then I will introduce you to the new 2.5 Duplex Assay. I will present the technical product configuration, the features and the benefits, and then I will present some data that we generated at ATD by our R&D team. So here is an overview of the RNAscope. So the in situ hybridization has evolved over the last 40 years. It has been established in the late 1960s as radioisotopic in situ hybridization, and then it evolved as a fluorescent-ish, and then there was plenty of improvement to the technology that was mainly meant to increase the signal over the background. The last improvement were done in 2012. And work published by Wang et al., and they describe the RNAscope technology. The RNAscope technology has increased sensitivity over traditional in situ hybridization and a decreased background. So I'm going to show you how the RNAscope technology works in more detail. So the RNAscope is a proprietary in situ hybridization technology based on a unique probe design and signal amplification. The platform has the sensitivity to detect most genes in situ as well as also several genes simultaneously. Just as a traditional-ish assay, the method starts by permeabilizing the cells of the tissue sample. Then you have hybridization of the probe, what we call the double Z probe, to the target RNA. Then the signal is amplified by a cascade of hybridization events. And then the signal can be visualized as single punctuated dots. Signal can also be quantified. So in more detail, this is how our probe design works. Our probe is designed as what we call the Z probe. The lower part of the Z hybridizes the target RNA over a sequence of 18 to 25 base sequence. Then there's a spacer sequence, and then an upper region of the Z is the 14 base tail sequence. Now, when the Z work as pair, when two Z's are bound adjacent to each other to the target specific binding site on the RNA of interest, it creates a preamplifier binding site of a 28 base region. The Z pair hybridizes all along the target of interest. A standard RNAscope probe is a pool of 20 Z pairs that hybridize over 1,000 base regions. So this is how it works. When your target RNA is hybridized by excuse me. When the probe hybridizes the target RNA, so the Z hybridizes, and then when two Z's are adjacent to each other, it creates a binding site for the preamplifier, and the preamplifier then an amplifier molecule will bind to the preamplifier, and then the label probe will bind to the amplifier molecule. Now, over the length of the target RNA, several Z pairs can bind, but because the signal is amplified only when three Z pairs are bound to the target RNA, if the RNA is degraded or there is RNA binding protein, the signal will still be visible, which makes the assay extremely robust and very sensitive. So, because when only one Z probe binds specifically to amplification of non-specific hybridization, and there's a very low to no background signal. So this RNAscope gives a high sensitivity with an extremely reduced background. So the RNAscope typical target probe is 1 kb, is over 1 kb, it's a 20 bit pair. It is also suitable for coding RNA and non-coding RNA. It will also be able to detect partially degraded RNA. The shortest target we can, the shortest probe we can design is would hybridize over a target of 300 base. That would be a 6Z probe. 6Z probes. We currently have over 8,000 catalog probes available. However, if the target of interest is not on our catalog yet, it can be designed in 2 weeks. So to design a specific target, your RNA of interest, you will need to provide the accession number or the exact sequence of your target. You can as well indicate the shell requirements if you want, for example, the probe to target a specific region of your transcript. Then our bioinformatics scientists will channel this information to the ACD alignment algorithm and will select the optimal sequences to ensure the target specificity and uniformity of oligonucleotide hybridization in the assay. The algorithm evaluates the melting temperature of each oligonucleotide in the pool to ensure that there is no cross-hybridization with non-target RNA under stringent conditions. So the assay is universal. We can actually design a target against any gene, any genome in all species, and it can be used on any tissue or cell sample. As I said, we have almost 9,000 probes today, and you can design any probe that you wish to have. The RNAscope assay is applicable to a wide variety of research applications. We have now over 450 peer-reviewed publications in high-ranking peer-reviewed journals in various research focuses at cancer, neuroscience, immunology, to name a few. So it is my pleasure to introduce the ACD latest product, the RNAscope 2.5 Duplex Reagent Kit. The kit was actually developed based on customer demand to replace the existing Duplex kit. It is a green and red combination using the HRP green substrate and the fat red. The kit is optimal to identify expression patterns for up to 2 targets and map co-expression of 2 targets within the same tissue. So here are the highlights of the RNAscope 2.5H Duplex assay. It is based on the dual 2.5 chemistry. Both channels are amplified to the same level. There is a green and red color combination. All reagents are ready to use, and it is very convenient to use if you're already using the singleplex, as the same pretreatment reagent will be required. The benefits are that you can visualize low expressing targets in both channels. Again, as I said previously, it has comparable sensitivity to the 2.5 singleplex assay. It is very easy to use. You don't need any additional optimization, and you get a sharper, crisper signal in both channels that is easy to quantify. So here is the list of the reagents that are in the Duplex 2.5 Duplex reagent kit. So as you have for the brown or the red kit, you have the protease treatment, the hydrogen peroxide, and the protease plus, the target retrieval buffer, and you have 10 hemp reagents, the fat red A and B, and the green substrate A and B, as well as the wash buffer. I'm going to tell you a little bit more about the 2.5H Duplex probes configuration. So on the C1 channel, the signal is HRP mediated. The color will be green. On the C2 channel, it's alkaline phosphatase mediated, and the signal will be red. The two channels are relatively comparable in sensitivity, and due to the property of the red color and per pathologist preference, it is recommended to put the lower expressor in the red C2 channel. Here I present the positive controls that we recommend. For positive control, we recommend the PPIB as C1 and the polar 2A as C2. Please note that there is a change in the duplex positive control probes in order to avoid signal masking of the PPIB signal. Signal masking of the polar 2A signal by the PPIB signal. For this reason, we recommend to have PPIB in red as C1 and polar 2A in, PPIB in green as C1 and polar 2A in red as C2. So as the negative control is always the same with DAB in both channels. So when you request target probes, your target probe C1 will be ready to use and the C2 will be 50x. And you will mix your C2 probe and the C1 probe before your assay. We have, with the launch of this new duplex assay, we have introduced a new intro pack for the new duplex user. We have a human intro pack and a mouse intro pack. The human intro pack contains the HELA control site, the duplex positive control for humans, the duplex negative control, the reagent kit, the extra wash buffer, the pens, and the vector mounting media that we recommend for this assay. The same is for the mouse duplex intro pack with the 3C3 control slides, the appropriate positive and negative control, the reagent kit, extra wash buffer, and also the pen and the vector mounting media. So, how to get started with the new duplex assay, the new 2.5 duplex. My personal recommendation is that new users of the technology who are not familiar with the RNAscope method should start with the red assay to get familiar with your RNAscope and get confidence and expertise with the single assay. We recommend that all new users of the duplex assay would start with the intro pack that I described previously. As for any RNAscope experiment, please ensure that you review the checklist for the 2.5 duplex assay. This one is available on the support page. And also review the user manual to familiarize yourself with the workflow before you start your experiment. So here is more details about the assay workflow. The 2.5 duplex assay is overall longer than the former duplex assay. The assay runs over about 13 hours and is easily split into 2 days. So usually for the single-plex assay, we have 3 recommended stopping points: after the baking, after the paraffinization, or after the target reducer step. For this new duplex assay, we introduced a 4th optional stopping point, and this is the stopping point we actually recommend, and it occurs after probe hybridization, overnight in 5 assays. The slides are placed overnight in 5 assays. So the first day of the assay is pre-treatment and probe hybridization, and the second day is the signal amplification and detection. The signal amplification and detection starts with the channel 2, with M1 to M6, followed by the fast-red chromogen detection, and then followed by the channel 1 signal amplification, M7 to M10, followed by green chromogen detection, counterstaining with hematocrit, drying, and mounting with vector mount. We recommend to only use vector mount as a mounting media, and this will ensure stability and sharp signal with both chromogens. The quantification of the duplex signal can be performed using the Halo software from INDICA. Each cell is assigned to a bin based on the number of dots per cell, and clusters are divided by the probe signal area to calculate dot number for a cluster. A H-score calculation is performed in Halo to assign a single score to an entire tissue section based on the average target expression of the tissue. If you would like more information about Halo signal classification, you can contact INDICA Lab directly. I can give you their contact information. So here is a summary of the key workflow highlights of the duplex assay. So we actually recommend to split the assay over 2 days with a stop after probe hybridization. There's an easy transition from the single-plex assay to the duplex assay because the pretreatment condition remains the same. We recommend that the lower expressor is placed in the SITU channel as the red color is visually stronger. And as I just said, the automatic quantification of the signal can be performed within the Halo software. So I'm going to show you now some application review with the data that we generated at ACD in California. So the RNAscope 2.5 duplex reagent assay kit has been tested in-house over 76 samples. We tested the assay in human TMA, mouse tumors, mouse tissue, and xenograft tissue. Here you can see an example of a positive control polar 2AC1 PTIBC2 in brain, in colon, and you distinguish the green signal as sharp green dots, obviously, and the red signal as well very sharp. Here are some examples about the target application we can have with the RNAscope 2.5 duplex assay. In neuroscience, you can use this assay to colocalize cell type marker to biological marker of interest. In PDX, patient-derived xenograft, you can evaluate human tumor and mouse trauma gene expression profile. This is an example of an expression of human PCAM and mouse PCAM in colorectal PDX tissue sample. I will show you more detailed examples later. In immunotherapy, you can use it to detect RNA targets in the tumor microenvironment. And in oncology, you can study the ligand receptor expression in the tumor microenvironment. Here again is another example of the 2.5 duplex assay in immune cells infiltrating lung tumor and ovarian cancer. In ovarian cancer, PD-L1 in green, CD8 alpha in red. You clearly see CD8 plus cells in tumor and stroma, and PD-L1 expressing cells are most likely immune cells in the tumor there. In the lung, on the right hand side, you can see a high abundance of CD8 plus cells in tumor and stroma, and some colocalizing with PD-L1 plus cells. Here's more presentation of the data. So FFPE tissues tested in-house with the duplex assay. We tested thoroughly cell pellets with LS cells pellets, the same pellets that you're getting when you're purchasing the IntraPack. We also tested numbers of human tissues, tumor, liver, tonsil, colon, breast, mouse tissues as well, and human xenograft in mice. Please note that so far the 2.5 duplex assay was only tested on FFPE tissue samples. Fresh frozen tissue samples tests are in progress. It has not yet been validated on culture cells and PBMC. So here is an example of a control run on hairless cell pellets. It's a control CBP, C1 polar 282. That B negative control, you see cell morphology well preserved. That B negative control is clear, and you see both signals appear as sharp concrete dots in both channels. Again, this is also an example of duplex 2.5 duplex assay you can clearly see green signal appears. Some cells are specifically express NGR1, while other cells express ARBB3. The signal is nicely distinguishable and is specific to certain cell populations. This is another example with actually much stronger signal. In kidney cancer and colon cancer, CD248 and PDG-FRB. This is a more, excuse me. This is another example of staining in mouse colon, mouse brain of LGR5 and B1. Here in higher magnification, it's very nicely distinguishable, the dot of the LGR5 staining versus the red dot of the B1 staining. This is a very beautiful staining. So here is another example of staining with the 2.5 duplex assay in patient-derived xenograft. Here we have performed experiments with, in the C1 channel, a human EGFR probe that doesn't cross-react with mouse, versus in the C2 channel, red, a mouse EGFR probe that doesn't cross-react with human. Here you see that C1 staining only occurs in the tumor area, while the mouse EGFR probe only targets the stroma. This is a similar example with PCAM, and C1 channel, a human PCAM probe that doesn't cross-react with mouse, versus in the red C2 channel and only staining the stroma, a mouse PCAM probe. You see that specific probes result in highly specific signal in the corresponding tissues. Again, another example of staining of CDX with 2.5 duplex assay with other probes, human FGFR1 probe that doesn't cross-react with mouse versus mouse FGFR1 probe that doesn't cross-react with human. Again, you see the green signal limited to the tumor area, whereas the mouse red FGFR1 is visualized in red in the stroma area. That's the same here on the right-hand side with TGF-beta1 versus humans in green versus mouse TGF-beta1 in red. So here I'm going to present some further data showing the technical detail and the reproducibility of the assay. Here, what has been done is that we have run several different slides using two different ovens and using a positive control probe that is CPD in the C1 channel and polar 2A. You see that there is similar results on different slides and also in different ovens, showing that the high reproducibility of the assay between different slides and different ovens. Here is the same, at different days. The assay was performed using the following the exact protocol on different slides and different days. And this is also performed by different scientists. The same as all those experiments were performed on cells. So here's the conclusion of what I described today. So ACD continues with the product innovation and is now introducing the new RNAscope 2.5 duplex assay. We are demonstrating a robust result in different tissue types and with different targets. This new assay was thoroughly tested in-house, demonstrating reproducibility within and between runs, different ovens, and also different operators. So, if you have any further questions, any technical questions, you can always contact the support team of ACD at support@acdbio.com. We have a direct phone number for support in the USA, and for support Europe, this is my cell phone number. is there is a tremendous amount of resources on our website, so I invite you to have a look at it and go through the support page where we describe how to get started and we answer most of the frequently asked questions. Thank you very much for your attention, and I will be able to answer some of your questions right now.